first thank the conference organizers. Profound gratitude for me to share the stage with many pillars in mitochondrial research. I know lunch is coming up, so I'll try not to take too much time, but obviously our group is interested in mitochondrial transplantation, and I'm going to take you through a story that is hopefully the beginnings of getting us there. Is targeting the brain is no straightforward thing, and so I'll talk about astrocytic mitochondrial transfer to brain capillaries. So just a quick disclaimer. As you just heard, Beilu has really driven this project forward in our group. So I'd like to thank him and the funding, and Dr. Sullivan as well, Dr. Patel, and the group that hopefully you can see we work pretty well together. So one of my major interests in brain health and brain restoration is the neurovascular network. Hopefully it's obvious that this is important. You have 500 miles of vascular network in the human brain, and so obviously dysfunction at this level can cause pretty widespread effects. And so the endothelial cells have been thought to be mainly glycolytic, and they possibly are, but I think there's a lot of recognition in the importance of mitochondrial function in the vasculature and endothelial cells themselves. And so a lot of neurodegenerative disease, as we've heard, is driven by oxidative stress and mitochondrial dysfunction. And so a lot of these processes, angiogenesis, tight junction repair, that we want to when you have an insult or when you have chronic disease, these are things that you want to restore. And hopefully the field agrees upon that these are mitochondrial processes. And so getting into this field, we're really just interested in, can we explore in vivo whether mitochondrial transfer occurs at the capillary level? And so this is difficult. It's not straightforward in vivo because you have all these cells that are part of the blood-brain barrier. You have astrocytic infeed, pericytes, endothelial cells in the endothelium with a tight junction, all these cells that are really close together. And it's hard to know really, is transfer happening based on the state of technology right now. We know that astrocytes, it's appreciated that they have a very high level of mitochondria compared to neurons. And so why is this? And we know that even the astrocytic infeed have mitochondria in them. So really we wanted to look at this using the same model that others have looked at, the mitochondrial 2 labeled model. We wanted to simply ask the question of, can we track transfer? So the idea is, how's the best way to do this since we have a complicated system? And so we've already heard a bit about these MitoD2 labeled mice. We utilize the global model, which expresses the green fluorescence in every mitochondria. And then we also use astrocytic specific model where mitochondria only fluorescent in astrocytes. And so we really manipulated this model. So we use this astrocytic specific model, and then we paired it with another technique in the lab that we use where we can actually pull out the capillaries, so vessels that are less than 10 microns width across. We pull these out, and we've shown that we can actually isolate the astrocytes away from these vessels. So what we pull out is endothelial cells and pericytes. And we've shown this here in the publication. And so we published this, I believe early this year in the journals through Blood Flow Metabolism. Kazu was the editor for a special issue. So all of this data is published, and I'll direct you to that. I don't think you can see the citation very well, but really just briefly going through this, what we saw is that using similar confocal and Imaris techniques, we were able to localize mitochondria that originated from astrocytes within the endothelium. So you can see that here in endothelial cells. You can see that next to the nuclei in the endothelium. This seems to be only in young animals, only a small portion of the total mitochondria, and so it's about 4 to 5% of the, or maybe closer to 2% of the total mitochondrial volume are these transferred mitochondria, but this seems to be happening. And hopefully you can appreciate and visualize that this is our global expressors, so every cell expresses, and these are the transferred mitochondria. And these also co-localize when we stain with the TALM-20 stain. And so we also see this being transferred to the pericytes, which are the smooth muscle cells of these smaller capillaries. And so we see a similar thing where you have co-localized with this cellular morphology, bump wall morphology, as you would call it, these pericytes. And so you can see these transferred mitochondria at this level. And so then we wanted to ask, we're interested in aging as one of the contexts of this. What happens in aging, and we potentially think that this is actually healthy aging. So these mice are 1 month and 5 months after we give tamoxifen, which expresses the fluorescent protein in astrocytes, but these mice are really 3 months and 7 months of age. And so we see this increase in astrocytic transfer to brain capillaries with what we think is healthy aging. And so we've already heard about compensatory effects of this transfer. This may be what's going on. There's still a lot of questions to answer. And then finally, we've heard about, what's the method of transfer and how can this actually relate to transplantation. And so we, I won't belabor the point, but we're looking at, extracellular vesicle transport. And so we performed an in vitro study and found that when we actually isolate extracellular vesicles with mitochondria from cultured astrocytes, we can incubate these with endothelial cells, and indeed we can see that there's transfer and that these mitochondria are active. And so we're showing that in this video here. I think you've already seen the top panel from Beilu's talk, but just to show again, these are transferred mitochondria using extracellular vesicles into endothelial cell culture. And we also have a cold culture where we also see the same thing, that here's an astrocyte here, but we have transferred mitochondria in endothelial cells coincident to where the astrocyte is. So, this is really burgeoning a new area for us, and we're interested in looking into the transfer further, but then also what does this mean as far as transplantation? Like we heard before, do we need brain mitochondria to target the brain? And so we're interested in where this would take us. I'm interested in brain trauma, so we're going to conduct experiments to see how traumatic brain injury can alter this process as well. So I'd like to thank the team again, and we'll take any questions.
Any questions from the audience?
Okay, sorry. One of the things that people have seen in Alzheimer's disease is a marked decrease in microvascular vascularity. Do you think that the changes we're seeing in Alzheimer's disease when you give mitochondrial transplantation might be actually affecting the microvasculature rather than, and therefore the perfusion rate rather than the actual numbers?
Yeah, that's a great question. It's something that we're very interested in. I think even maybe someone from Dr. McCauley's group, I saw angiogenesis on one of the posters back there, and that may be a actual biological target of this mitochondrial transplant. And it's something, it's more difficult to answer that question in the brain, but it's something that we're really dedicated to answering. Mitochondrial drugs, can they get into endothelial cells and increase these processes? And so, if you're of the belief, and I think many of us are, that mitochondria are at the center of these diseases, they may precede other things like vascular dysfunction. Can we target that, and can we look at that? And so, yeah, we'll definitely be looking at that in our model. So it's a great point. Thank you.
Yeah, Lance Becker from New York. This is wonderful because it's such an important question, right? And my question is just really quick, which is, have you followed your mitochondria to see where they go more? So like, if you just shoot them in, are they in the endothelium or are they getting into other cells? Where do they go really big question because it has to do with Doug's question, and I think you've got the ability to answer.
Yeah, that's definitely our next steps. We're funded to do that work. So all the vesicle EV mito, which we actually changed the term based on the recommendation of the mitochondrial, how we talk about these things. So these EV mito studies we've only done in cell culture up to this point, but we have funded studies where we're going to do it in vivo. And yes, we want to track it to see where it goes. So yeah, you're right on the next steps. Thank you guys so much.